Every living thing, from the smallest bacterium to the largest whale, starts with a single fundamental building block, the cell. This profound understanding forms the bedrock of biology and is a central focus of Professor Vanessa's work on the structure and function of cells. Let's begin with cell theory, which lays out three core principles. First, all living things are composed of cells. Second, a single cell is the smallest unit that exhibits all the characteristics of life. And third, all cells come only from pre-existing cells. Cells are classified into two general types based on their internal organization, prokaryotic and eukaryotic. While all cells share a plasma membrane enclosing them, their internal environments differ significantly. Prokaryotic cells are considered more primitive. Their internal environment isn't divided into membrane-bound compartments. In contrast, eukaryotic cells have an internal environment that's highly organized, divided into numerous membrane-bound compartments called organelles, each with specialized functions. Prokaryotes, on the other hand, include all organisms in the domains bacteria and archaea. They lack a nucleus and membrane-bound organelles. Their structure consists of a plasma membrane, often covered by a rigid cell wall, and cytoplasm. Crucially, their genetic material isn't enclosed by a membrane. Generally, prokaryotic cells are also much smaller than their eukaryotic counterparts. A fascinating aspect of cells is how their structure directly reflects their function. Even among eukaryotic cells, which are remarkably similar, there are distinct structural differences. All cells perform essential metabolic activities like gathering raw materials, excreting wastes, synthesizing macromolecules, growing and reproducing, but specialized functions lead to specialized forms. Think about muscle cells, which contain numerous organelles to provide the energy needed for contraction, or nerve cells, which are long and thin to efficiently carry impulses over significant distances. Every cell has some sort of specialized function, and its structure supports that role. There's also a critical reason why cells remain small: efficiency. The total metabolic activity of a cell is proportional to its volume of cytoplasm. All raw materials must cross the plasma membrane, and all waste must be expelled through it. As cells get larger, their volume increases much more rapidly than their surface area. This means small cells have a higher surface-to-volume ratio, which promotes efficiency in acquiring nutrients and disposing of waste. Some cells even develop small projections of their cytoplasmic membrane called microvilli to further increase this vital surface area. To visualize and study these tiny structures, we rely on microscopes. A light microscope can magnify objects up to 1,000 times and allows us to view living samples. For greater detail, transmission electron microscopes can magnify up to 100,000 times or more, revealing the intricate internal details of cell structure. And for a three-dimensional view of the cell surface, we use a scanning electron microscope, which can magnify up to 100,000 times as well. Now, let's look at some of the key internal structures within cells and their specific functions. The nucleus controls the cell and serves as its information center. Ribosomes are responsible for synthesizing proteins. The endoplasmic reticulum acts as a manufacturing center. The Golgi apparatus refines, packages, and ships cellular products. Vesicles are membrane-bound containers for storage and shipping, and mitochondria, often called the power plants of the cell, provide energy. The nucleus, as the control center, contains the cell's genetic information, or DNA, and regulates all cellular activities. Structurally, it's enclosed by a double-layered nuclear membrane, which features nuclear pores that permit the passage of RNA and proteins. Inside, we find chromosomes, which are the DNA, and a nucleolus, the site where ribosome components are synthesized. Ribosomes, composed of RNA and protein, are the sites of protein synthesis. They exist in two forms: free ribosomes, which float in the cytoplasm and synthesize proteins for immediate use within the cell, and bound ribosomes, which are attached to the outer surface of the endoplasmic reticulum and synthesize proteins destined for other organelles or for export from the cell. The endoplasmic reticulum, or ER, is a highly folded membranous network, and it comes in two types. Rough ER has ribosomes on its surface. These attached ribosomes manufacture proteins that may be modified within the ER, especially those to be secreted from the cell. Smooth ER, which lacks ribosomes, is involved in lipid synthesis, including some hormones, and the packaging of proteins and lipids for delivery to the Golgi apparatus. The Golgi apparatus then refines these synthesized products. It acts as the cell's packaging and shipping center, taking products, packaging them into vesicles, and shipping them to other locations within the cell or to the cell membrane for export.
Vesicles are essentially membrane-bound storage and shipping containers, and there are several types. Secretory vesicles contain products destined for export from the cell. Endocytic vesicles contain substances imported from the external environment. Peroxisomes contain enzymes that detoxify wastes produced by the cell, and lysosomes house powerful digestive enzymes. Mitochondria are truly the power plants of the cell. The number of mitochondria varies depending on the cell's energy requirements. Each mitochondrion is surrounded by a double membrane, with the inner membrane being highly folded. This folding divides the mitochondrion into an inner compartment and an outer compartment. This is the site of cellular respiration, a process that utilizes oxygen and produces carbon dioxide, ultimately generating ATP, which is a quick source of energy for the cell. Beyond immediate energy production, cells also store energy. Fat, specifically triglycerides, serves as long-term energy storage in animals, typically found in the cytoplasm of fat cells. Glycogen, a carbohydrate, provides short-term energy storage in animals, stored in the cytoplasm of muscle and liver cells. These sources aren't enclosed by membranes, but provide vital energy reserves. For support and movement, cells rely on the cytoskeleton, an internal scaffolding that helps maintain cell shape. It's composed of microtubules, which are tiny hollow tubes of protein, and microfilaments, thin solid fibers of protein. Together, they form a framework that supports the cell, much like tent poles support a tent, and they also anchor other cellular structures.
Specialized structures like cilia and flagella enable movement. Cilia are short and numerous, found on cells lining airways and certain ducts, where they help move substances. Flagella are long and typically single, like those that enable spermatozoa to swim. Cilia and flagella share a similar internal structure. Additionally, centrioles, short rod-like microtubular structures near the nucleus, play an important role in cell division. The plasma membrane is the boundary that separates a cell from its environment. It's selectively permeable, meaning it permits the movement of some substances into and out of the cell while blocking others. This selective nature also enables the transfer of information between the environment and the cell. Structurally, the plasma membrane is a lipid bilayer. It's primarily composed of phospholipids, arranged in two layers with polar heads facing the outside and the cytoplasm, and nonpolar tails meeting in the center. Cholesterol increases the membrane's mechanical strength. Proteins embedded within or spanning the membrane provide means of transport for molecules and facilitate the transfer of information. The membrane isn't rigid. It's often described as a fluid mosaic, where phospholipids and proteins aren't anchored and can drift relative to each other. Molecules cross the plasma membrane in several ways. Passive transport doesn't require the cell to expend energy. This includes diffusion, osmosis, and facilitated diffusion. Active transport, however, requires the cell to expend energy. Finally, bulk transport involves membranous vesicles to move larger substances through processes like endocytosis and exocytosis. Passive transport relies fundamentally on diffusion, the movement of molecules from a region of high concentration to a region of low concentration. This requires a concentration gradient, a difference in concentration between 2 points. Molecules move until equilibrium is reached, where the concentration is uniform. Diffusion is only effective over short distances, such as across membranes or within cells. Osmosis is a special type of diffusion, the diffusion of water across a selectively permeable membrane. Water moves from an area of low solute concentration to an area of high solute concentration, essentially moving down its own concentration gradient. Osmotic pressure is the fluid pressure required to exactly oppose this movement of water. Most substances cross cell membranes via passive transport processes, moving down their concentration gradient. There are three main forms. First, direct diffusion through the lipid bilayer, which allows small, uncharged, nonpolar molecules like oxygen, carbon dioxide, and urea to pass directly without assistance. Second, diffusion through protein channels, where proteins span the membrane, allowing specific molecules through based on the size, shape, and electrical charge of the channel's lining. Some channels are always open, while others are gated. Water and ions are examples of substances transported this way. Third, facilitated transport, also called facilitated diffusion. Here, a specific molecule binds to a membrane transport protein, triggering a change in the protein's shape that carries the molecule through the membrane bilayer. These transporter proteins are highly selective for specific substances like glucose and amino acids. Active transport is different because it moves substances from an area of lower concentration to an area of higher concentration, essentially moving against the concentration gradient. This process requires a membrane protein, often called a pump, and an energy source, typically ATP. A classic example is the sodium-potassium pump, which transports 3 sodium ions out of the cell for every 2 potassium ions brought into the cell, moving both ions against their respective concentration gradients. For moving larger molecules, cells use bulk transport mechanisms. Endocytosis brings substances into the cell. A substance in the extracellular environment is surrounded by the cell's plasma membrane and internalized, forming a membrane-bound vesicle. Some vesicles are selective and have receptors for specific substances, like the transport of insulin into cells. Exocytosis, conversely, expels substances from the cell. Here, a substance contained within a membranous vesicle fuses with the plasma membrane, releasing its contents to the external environment, such as in the release of wastes or the secretion of special products. The plasma membrane also facilitates the transfer of information. Certain molecules can influence intracellular activities simply by contacting the cell's outer surface. Receptor proteins span the membrane and are crucial for transmitting information to and from the cell. These proteins have receptor sites that interact specifically with signal molecules. For instance, an insulin receptor protein will only respond to insulin. When a signal molecule binds to its receptor site, it triggers a change within the cell. Different cell types, of course, have different receptor proteins. The sodium-potassium pump also plays a vital role in maintaining cell volume. It expels unwanted ions, stockpiles needed ones, and stabilizes the cell size. ATP is used to expel 3 sodium ions for every 2 potassium ions brought into the cell. An increase in cell volume, meaning more water in the cytoplasm, can be managed by decreasing the pump's activity, allowing more sodium inside. Conversely, to decrease cell volume or reduce water in the cytoplasm, the pump's activity increases, expelling more sodium ions. This leads us to the concept of tonicity, which is the relative concentration of solutes in 2 fluids. An isotonic extracellular fluid means the extracellular and intracellular solute concentrations are equal. In this state, cells maintain a normal volume. Regulatory mechanisms in the body work to maintain an extracellular fluid that's isotonic with the intracellular fluid. However, variations in tonicity can occur. In a hypertonic solution, the extracellular solute concentration is higher than the intracellular concentration. Water will diffuse out of the cell via osmosis, moving towards the higher solute concentration, causing the cell to shrink and potentially die. In a hypotonic solution, the extracellular solute concentration is lower than the intracellular concentration. Water will diffuse into the cell via osmosis, moving towards the higher solute concentration inside the cell, which can cause the cell to swell and burst.
Cells are incredibly dynamic, constantly using and transforming matter and energy through metabolism, which is the sum of all chemical reactions in an organism. Thousands of these reactions occur concurrently, often organized into complex pathways that can be linear, branched, or cyclic. We can categorize metabolic processes into 2 types: anabolism, which builds larger molecules from smaller ones and requires ATP input, like assembling a protein from amino acids, and catabolism, which breaks down larger molecules to smaller ones and releases ATP output, such as breaking down glucose. Every chemical reaction within a cell requires a specific enzyme. The metabolic activities of a living cell demand a lot of energy to build large, complex macromolecules, power active transport, and fuel movement like muscle contraction. This energy is often provided by the catabolism of ATP, which acts as the cell's energy cache. Glucose is a primary source of this energy. The energy stored in glucose is used to generate ATP, with one glucose molecule potentially yielding about 36 ATP. In the absence of glucose, other carbohydrates, fats, and proteins can be catabolized to generate ATP. ATP itself is a more readily used pocket change form of energy. Glucose and other fuel molecules must be cashed in for ATP, which can then be used to perform cellular work.
The breakdown of glucose in the presence of oxygen to yield ATP is known as cellular respiration. This complex process occurs in 4 main stages: glycolysis, the preparatory step, the citric acid cycle, and finally, the electron transport system and oxidative phosphorylation. Glycolysis, the first stage, occurs in the cytoplasm. It's a series of 10 reactions that split glucose into 2 molecules of pyruvic acid, or pyruvate. This stage requires an investment of 2 ATP, but it produces 4 ATP, for a net yield of 2 ATP. During glycolysis, high-energy electrons and hydrogen ions are also removed and picked up by a coenzyme, forming NADH, which will be used later to generate additional ATP. Next is the preparatory step. The 2 pyruvate molecules from glycolysis enter the mitochondria. They are converted to acetyl groups, releasing carbon dioxide. High-energy electrons and hydrogen ions are again removed, picked up by a coenzyme to form more NADH. These acetyl groups are then joined to coenzyme A to form acetyl-CoA, which will enter the citric acid cycle. The citric acid cycle, also known as the Krebs cycle, harvests more energy. It occurs in the inner compartment of the mitochondria and is a cyclic series of 8 reactions that break down the acetyl-CoA. The molecule, oxaloacetate, that accepts the acetyl group from acetyl-CoA is regenerated at the end of each full turn of the cycle. The cycle extracts high-energy electrons to form NADH and FADH2, and it produces 2 ATP and 4 carbon dioxide molecules per glucose molecule.
The final stages are the electron transport chain, or ETC, and oxidative phosphorylation, which produce the bulk of the ATP. The ETC is located in the inner mitochondrial membrane, separating the inner and outer compartments of the mitochondria. It consists of many electron carriers arranged in the membrane. It takes electrons from the NADH and FADH2 produced during glycolysis and the citric acid cycle, passing them down a chain of electron carriers. The energy lost by these electrons is transferred to the carriers. The final electron acceptor in this process is oxygen. Oxidative phosphorylation directly uses the energy released by the movement of electrons down the ETC. This energy pumps hydrogen ions from the inner compartment of the mitochondria to the outer compartment, creating a gradient. There's a higher concentration of hydrogen ions in the outer compartment and a lower concentration in the inner compartment. An enzyme called ATP synthase embedded in the membrane allows these hydrogen ions to flow back across the membrane down their gradient from the outer to the inner compartment. This flow fuels the synthesis of ATP from ADP and inorganic phosphate. This entire process is known as oxidative phosphorylation.
In summary, cellular respiration involves over 20 different enzyme-catalyzed reactions. It releases the energy in glucose slowly, capturing that energy in the bonds of ATP. Approximately 36 net ATP molecules are produced from each molecule of glucose. In this process, oxygen is consumed and carbon dioxide is produced. Cellular respiration is truly the cellular process that uses oxygen and produces carbon dioxide in the complex process of making ATP.
Beyond glucose, fats and proteins serve as additional energy sources. our total energy reserves are roughly 1% glycogen, which can rapidly catabolize to glucose and then enter cellular respiration. Fats, specifically triglycerides, constitute a massive 78% of our total energy reserves and have twice the energy of an equal amount of carbohydrates. Proteins make up about 21% of total energy reserves and have the same amount of energy as an equal amount of carbohydrates. Fats are broken down to glycerol and fatty acids. Glycerol can be converted to glucose in the liver or to pyruvic acid, which then enters the citric acid cycle. Fatty acids are broken down into 2-carbon acetyl groups that enter the citric acid cycle as acetyl-CoA. Proteins are broken down into amino acids. Amine groups are removed from these amino acids, then converted to urea and excreted. The carbon backbones of the amino acids can enter various points of the citric acid cycle. While proteins can be an energy source, their primary role is as enzymes or structural components. Protein catabolism typically increases during starvation.
Finally, cells have anaerobic pathways to make energy available without oxygen. If oxygen isn't present, cellular respiration can't continue. Glycolysis will still occur, but pyruvate will build up and be converted to lactic acid. This lactic acid buildup in muscles can cause a burning sensation. Anaerobic metabolism yields a limited ATP production, typically only 2 ATP per molecule of glucose, compared to the 36 ATP from cellular respiration. Once oxygen becomes available again, the lactic acid can then be metabolized aerobically. Professor Vanessa's comprehensive overview truly highlights the incredible complexity and elegance of cellular life, from tiny components to vast energy transformations. Consider sharing this episode with someone curious about the fundamental units of life.